It wasn't until a few years ago that my interest in actually getting my field IDs right was sparked mainly by Peter Cowman, who put in an application, a grant application, to put together a phylogeny of the Acropora. Now, I read his grant application. I said, "You're crazy, crazy, Pete. This is just not going to work. It's just not possible to do this in the time frame." But luckily, the reviewers disagreed or didn't know their stuff, and he got the grant. And so we had a license to go and some money to go and try and collect. Pete's ambition was to collect every Acropora species there was. And, you know, given that they're found from the Red Sea all the way to Hawaii, from Japan all the way down to the Solitary Islands, like, again, crazy ambitious program, but we've been trying. And it's been a wonderful excuse to travel, but you know, forcing me to look more closely, and again, introducing me to these molecular techniques, which is again not in my training. It's really opened my eyes to the fact that the taxonomic framework that we've been working with for the last 20 years is fundamentally flawed. It's unworkable, but there is a way forward. You know, the tools are there now for us to make some real progress. And so there's the background. And so, I mean, and again, lots and lots of people have been involved in this project, Tom Bridge and David Miller as well. So anyway, so the talk outline, I'm going to give you a brief introduction to 250 years of coral taxonomy, including the crash course in taxonomy that I wished I'd been given as a biologist and an ecologist when I did my undergrad degree at James Cook University back in the 80s and 90s. I'm going to present the Veron framework, which is essentially the framework that we all work with or try to work with, and subsequent tests of its validity. And then I'm going to present some case studies of, you know, when taxonomy matters. And the case study I'm going to focus on is looking at the taxonomic identity of 22 of the Acropora genomes that have been published. So most of our data is on the Acropora. And so, you know, we can harvest these genomes and of the markers that we're using and look at the identity compared to our specimens. And then I'm going to talk about the way forward. And if I've got time, I might just briefly explain why I'm here on this JSPS fellowship. So taxonomy started, or modern taxonomy started with Linnaeus in 1758. And this is the first, one of the first Acropora species ever described. And so on the left, you can see Linnaeus's original description, a couple of lines in Latin. And then there's the drawing that he based his description on, which is a specimen collected by a Dutch naturalist called Rumphius from somewhere in central Indonesia. That specimen was subsequently lost, if it ever existed. So when Carden Wallace did her revision of the Acropora in 1990, she designated a neotype. And so that on the right there is the neotype of Acropora muricata. And you can see it's a very good representation of that image. Now, after Linnaeus, you know, over the next 230 years, you know, up into the late 80s, people from all over the world, many different countries, just started essentially just describing new species. You know, it was all the rage to document the biodiversity. You know, and again, with different people working on different continents, it was inevitable that on occasion, the same species would be described more than once. You know, and so, you know, rules evolved to work out how to deal with that. Now, but essentially over those 230 years, it was essentially a steady accumulation of new species, new nominal species. There were some attempts to address those issues where the same species had been given two separate names, but this was the first major revision of that 230 years of Scleractinian taxonomy. It was a work, 5 volumes over 8 years, mainly led by Charlie Veron. focusing on the Sclerotinia of Eastern Australia. All their collections were done there, but they did make an effort to examine much of the type material. And so in this work, when they started, there were 230 nominal species, 230 species, 2,300, sorry, nominal species had been described. And they reduced that number down to 700 species that they thought were good species. So what we call accepted species. So there's 1,600 synonymies and also taxon inquirendum. These are species they couldn't work out what to do with. But so there's 1,600 synonymies contained in that work. Now, and so again, once you describe a nominal species, that name is out there forever. It doesn't change. And so now this is just to describe the process of synonymization. If you come along, another later author comes along and thinks that these two different nominal species are actually one real species, they synonymize it. And I'll just give you a quick example. This is on the left there is Acropora millipora, which was described by Ehrenberg in 1834. On the right is Acropora spathulata, and it was described by Brooke. It's from the Solomon Islands. We don't know where Acropora millipora is from. Now, based on the morphologies, just looking at the morphologies, or probably even just photos of the morphologies, Veron and Wallace in 1984 decided that millipora and spathulata were the same. So they synonymized the later name on the basis of priority. So that one of the rules of the taxonomic code is that the earlier name wins in questions of synonymy. And so, and then in 1994, Carden Wallace and Betty Willis were doing a lot of work on hybridization on the Great Barrier Reef. And they realized that these two different morphs of millipora were not interbreeding. And so they decided that therefore this was good evidence that they were actually two distinct species. And so they resurrected the name spathulata that had been synonymized previously. And so you can see that the number of names, different categories of name changes through time. In 1834, we've got the one nominal species. In 1891, we've got two nominal species. In 1994, we've still got those two nominal species. They never go. But one's considered valid, one is synonymized. Then in 1994, we're back to the case with two nominal species, both valid. So what's happening, behind the scene in terms of, the accepted species is that there's this huge number of species that are not making it into the guidebooks. So here's a graph. The red line is showing the number of nominal species through time. And you can see that it just continues to climb as new species are named. It comes in bursts. Dana was one of the first to describe lots of different species. Another burst was Brooke in 1891. Veron and Wallace, you can see that they did describe some new species, but because of all their synonymies, the number of accepted species declined quite drastically after their work. Again, slowly climbing again until Veron 2000. And, Charlie named a whole heap of new species in 2000, so the number of nominal species jumped. But then Carden later synonymized a number of Charlie's new Acropora species in particular. So again, number of nominal species climbing through time, the number of accepted species going up and down. And a strong element of personality involved in the decisions. So when I started my biology and ecology as an undergrad at James Cook University, this was the book that we used, the Corals of Australia and the Indo-Pacific. Now that book only contained about 600 species, I think, and none of us, we weren't aware of the turmoil that had gone on behind that. We weren't aware that there were 2,000 species names that weren't even mentioned there. And again, this is in 2000, Charlie put out Corals of the World, in which he presented, his hypothesis of the taxonomy of the Sclerotinia. And 799 species named in this book, 112 genera, 18 families. Not only did he put out that sort of taxonomic framework, he also put out maps showing the distributions of every single one of those 799 species. And here's an example, just one, Acanthasteria baobanki. And he also put out a theory as to the evolutionary relationships amongst these 18 extant families and a whole heap of extinct families as well. So that was the Veron framework presented to us in 2000, and that we've mostly been working within for the last 20 odd years. Now, a couple of important points to make about that framework. There was no molecular data that went into Veron 2000. No quantitative morphology, no reproductive biology. So, morphology was the one line of evidence that they used. No experimental tests of key concepts such as morphological plasticity. And, and again, what I found out over the last couple of years of re-examining all the type material is that they often didn't look very closely at the type material. And so there have been a number of tests. Certainly the advent of molecular data, the first serious challenge to the Veron hypothesis emerged. And this was work by Hiro Fukami in 2004. And this is the tree, the phylogeny that Hiro produced. Now, different families, species of different families are marked by different colours. And so, for example, the green one is the one that stands out nicely. These species in this family are scattered throughout the tree. And you can see that in most cases as well. And so of those 18 families that Veron hypothesised in 2000, not a single one of them was proven to be monophyletic. Very few of the genera as well. And this is my favourite example. This is the genus Montastria. So Charlie thought there was 12 species in this genus. Subsequent work by people all over the world has shown that these 12 species, well, they're now in 6 different genera in 3 different families. So, I mean, again, that's how poor morphology is as a guide to phylogeny in some of these genera. This is, testing Charlie's ideas on biogeography. So what I've got done here is I've listed the 20 most widespread coral species based on Charlie's data at Coral Geographic, and where Charlie defines ecoregions. So I've ranked them in terms of the number of ecoregions that they occur in, and also by the extent in square kilometres of their range. And you can see that of those 20 species, not all of them have been examined, but every one of them that has been examined has been demonstrated to be species complexes. So for example, the top, Postlepora damicornis, what Charlie's idea of Postlepora damicornis is at least 4 or 5 species. Probably the most interesting one is Hyacinthus. Charlie's idea of Hyacinthus, we think, is probably now closer to 20 different species. So rather than one widespread species found from the Red Sea all the way out to French Polynesia, it's about 20 different species. So, the biogeographic data is very flawed. Okay, now here's some of the data that we've done, some of Pete and Tom and my work. So testing, how good was Charlie's taxonomy of the Acropora? So he recognised 166 species. There's 412 nominal species. So there's 246 nominal species that are not mentioned in corals of the world due to synonymy or being unresolved. So this is a tree that we're hoping to publish very shortly with 890 specimens. We've got, within those 890 specimens, we've got 272 what we call primary species hypotheses. And these hypotheses are based on the morphology, looking at the type material and the molecules, obviously. So there's this interesting 6-clade structure. And of those primary species hypotheses, only 50 of the 272 can we match to the type material. 28, I'm sorry, that's not true. We can also, 28 of these we can match to the type material, and they're species that have been synonymized. There's 122 that doesn't look like any of the type material, but there's not necessarily good enough evidence to determine them as new species. And there's at least 72 that we're very confident are undescribed. So, there's an extraordinary amount of hidden diversity out there. It's hidden because species have been undescribed, and because they've been synonymized. So only 50 of the 272 of our primary species would you see in corals of the world. So, you know, whenever you go out onto the reef, it's no wonder that you can't put names on things, because they're all new or hidden. So yeah, we think the number based on our data, and we've since expanded that tree to nearly 2,000 specimens. Yeah, we think that the true number, the true species richness of the genus Acropora is probably closer to 400 or 500 rather than the 166 recognised by Veron. The other thing is that almost all the synonymies we have tested are incorrect. And there are just dozens and dozens of new species out there. So this is just an example from Sissoko. Now, I think within what people have been calling Hyacinthus at Sissoko, there's at least 3 species. In fact, you know, work from Go Suzuki and others has shown that there's probably at least 5 different species of what people have been calling Hyacinthus around Okinawa and throughout Japan. Okay, so what are the implications of a flawed taxonomic framework? This is a list of about 20-odd papers produced by the Centre of Excellence for Coral Reef Studies at James Cook University. All of these papers, many of them in Science and Nature, Ecology letters, are based on Veron's biogeographic data. So it means every single one of those papers needs to be revisited. Here's a specific example from one of those papers. This was one of Terry's papers in Science in 2003. This is a heat map of the world showing average temperatures at different locations. And then the contours are the number of species that are shared in common between regions. And so you can see that Lord Howe Island and the Gulf, Persian Gulf, have 24 species in common. The average annual sea surface temperature at Lord Howe is 24 degrees C. The average annual sea surface temperature is 36 in the Gulf. That sounds a bit high, but anyway, there it is. We argued that there were 24 species could withstand that huge range in temperature. And therefore, we argued that, you know, all was not lost for the world's reefs. There was substantial capacity for species to evolve to local conditions. We now know that, again, certainly there's not a single Acropora in common between the Gulf and the Great Barrier Reef. There's probably not a single Acropora in common between the Red Sea and the Barrier Reef. So, and even Lord Howe Island, only about 30% of the Acropora on Lord Howe Island occur on the Great Barrier Reef. So our conclusions were obviously wrong. This is a study by Gomez Corrales and Prada. What these guys did was they reanalyzed data looking at the thermal tolerance of Orbicella fulviolata, data from an earlier paper. The earlier paper had assumed that all these colonies were from one species. These guys reanalyzed, had another look at the genetics and determined that they're actually from 4 different clades, and the clades are coloured by blue. Now, the earlier paper concluded that there was sufficient variation in thermal tolerance within the population for this species to adapt to a 2-degree increase in temperature as a result of climate change. When these guys reanalyzed the data as 4 different species, they demonstrated that there wasn't, not in a single one of those different genetic clades was there sufficient variation for evolution to occur at that speed. So, by taking account, by using them, if you ignore the taxonomy here, you get exactly the opposite answer. This is an interesting paper from the Palumbi lab by Sheets et al. Now, and they showed that, if you want accurate estimates of connectivity, you need to be really careful about your taxonomy. So they've got a system where they define, I think it's 8 different clades of Hyacinthus cytherea, of the Hyacinthus cytherea species complex. And, it's just done at the regional scale and at the reef scale. So they've got data at 2 spatial scales. And they showed that on the x-axis, you've got what they call the true FST, which is the population value if you look at it as 8 different species. And on the other axis, it's if you just look at it as one species. And you can see that if you look at it as 8 separate species at the local scale, you overestimate FST, which means you're underestimating connectivity. And the opposite occurs at the larger scale. So it's not, it's scale dependent as well as, highly dependent on whether you analyze that as one species or 8 separate species. And a final example here is, again, from Japan. And so back in 2011, Yamano and colleagues published a paper showing that some species of tropical corals appeared to be on the move, in particular, Acropora hyacinthus. And, very good data going back 50 or 60 years, showing that areas in northern Japan, in Kyushu, there's been a phase shift from kelps to corals. No question that that's happening. The data's good, but Go Suzuki and colleagues had a closer look at these Acropora hyacinthus populations, and they've got 5 different genetic clades there, which are almost certainly 5 different species. And interestingly, the only one of those species that's on the move is the temperate population, the northernmost population. So this isn't tropical corals moving into northern Japan; it's temperate corals expanding their range. So again, sure, big changes, but not quite the tropicalization that people suspect. And yeah, it's very similar things are happening in Australia as well. It's the subtropical species that are doing well and moving down the coast, whereas the tropical species don't appear to be on the move as yet. Now, and there are lots of cases where taxonomy isn't important. There's a number of case studies where it is. And yeah, if you just after a representative genome of a coral to compare to a fish, it doesn't matter what the taxonomy is. If you want to compare a Montipora to an Acropora, you don't need to know the species identity. In fact, most organism biology, where you're looking at mechanisms rather than wanting to compare among species, you can get by it. It doesn't really matter what you call Montastria orbicella, just as long as you're only looking at Montastria orbicella. And so there are lots of questions that are independent of species identity. And, so this is not a threat to most of the experimental biology we do. It's just something we need to be aware of. It is a threat to biogeography. I mean, realistically, there are a number of things that are just not going to be possible to do for a number of years. Okay, so here's our test of the identity of the genomes. And so Pete went online, grabbed the genomes, and we can harvest the same markers that we use in our ultra-conserved elements. And so we can put those genomes into our phylogeny. And again, our phylogeny is informed by topotypes, which is not something I've talked about now, but one of the main things, one of the main advantages, I believe, of our approach is that we're revisiting the type locations of all of the nominal species. And so we collect a specimen that looks as close to the type as possible. So we know what hyacinthus looks like. So we know what Cytherea looks like. And then we can compare them molecularly. And so those topotypes are the anchor of our tree. And if we don't have a topotype, then we don't claim to know what that species looks like. Yes, and where there were vouchers, we also looked at them. Okay, so this is just one small part of that tree. We've got a tree with all 22 genomes in. But this is just the sort of, I think this is clade 6, the hyacinthus Cytherea clades. Now, in blue are the genomes. There's 3 Acropora 3 hyacinthus genomes and 1 Cytherea genome. And in black are our topotypes. The hyacinthus up there from Fiji, Cytherea down here from Tahiti. So you can see that there's 3 hyacinthus genomes. They're almost certainly 3 different species, and none of them are hyacinthus. And again, it's like the Cytherea genome is almost certainly closer to hyacinthus than it is to Cytherea, which I don't think I don't think Cytherea occurs in Japan. I'm not sure Hyacinthus does either. But look, so, we can go through the whole tree. That's just one example. And yes, so there we go. And again, not only are they not Hyacinthus, we think they're all undescribed. So again, it's impossible to put correct names on these things if they're not yet in the books. And so here's a summary of those 22 genomes. I won't go through them one by one. I'll just summarize it here. Probably only 4 of those genomes are correct, and that includes the 2 Caribbean species. And there's only 2 species in the Caribbean, or maybe 3. 3 are possibly correct. Again, we don't know without the topotypes, so there's a limit to how informative our phylogeny is until we get all the topotypes. But 15 of them are definitely incorrect. They just don't match the type material, or they appear in different parts of the tree. Okay, so of those genomes, 9 are likely to be undescribed, 6 are synonymized, and only 6 of them are accepted. So only 6 of those 22 genomes could you use Veron's Corals of the World to identify. So I'll just quickly show you a few examples from Okinawa. I presented this last time at Sissoko, and so all of these corals were just out the front, and with lots of people working on them. Oh, that's not working. Right, I won't worry about that. I should have. Okay, but look, so we've got 32 samples in our tree from Sissoko, and we think there's 14 primary species hypotheses there. 5 of them are accepted, 6 are undescribed, 3 synonymized. So this is what we're seeing everywhere we go. Only 1/3 of the corals in any one location are in the books. So again, I think these examples highlight the fact that the current taxonomic framework is fundamentally flawed. Most of the synonymies are incorrect, and we haven't even begun to describe the diversity that's out there. And so again, there's taxonomy moves. I've heard someone told me the other day, Kathy McFadden, who's a soft coral taxonomist. She told me that taxonomy proceeds one death at a time. It's really hard to move the field on while the authority figures, the gatekeepers, still control it, and that's been a real problem, I think, in Acropora taxonomy, in Sclerotinian taxonomy. But so it really is. It's well past time to ditch the accepted taxonomy. We need to move on. We need to look at this with fresh eyes. We need to resurrect and re-examine all of the synonymies. We need to test all of the nominal species with topotypes. So that's the way forward. This is our what we call our endeavour, Project Phoenix, and there's a number of things that we're doing. We're curating the type material, and we plan to make it freely available. Indeed, happy to give it to anyone if they want it. And that's involved visiting most of the museums that house coral specimens. So that was a 3 to 5 year project to get that happening just for the Acropora, mind you, Acropora Day. We've only just begun to expand to look at the entire order, and collecting the topotypes. And again, that there's a lot of work going out, finding them. It's why I'm here in Japan, actually, is to get topotypes for all the nominal species here, and then use an integrated and quantitative approach that combines molecules, morphology, ecology. So do it rigorously. 230 years of opinion really didn't produce much of value. So it's time to move beyond that. Again, I wouldn't despair. It's possible. You just need to be conscious to document the species that you're working with well, and that's easy enough to do. Good photographs, voucher specimens, tissue. And even if you can't identify it today, you can identify it down the track. So again, I think it's just really important to document, probably every animal that you use in your experiments. So you can go back and have a look. If you're getting some strange results, you can go back and tease apart some of those, some of that variation. Okay, and just quickly, yep, my JSPS fellowship was so that I could come out here and collect topotypes of the 64 nominal species that have a type location in Japan. Of those 64 nominal species, 26 were synonymized, and 11 are taxon inquiendum. 2 are not listed. So that only 21 of the 64 named here are currently accepted. My guess is that almost all of them are good species. The second aim of the project is species discovery too. So, as I say, there's 30% of endemicity is typically what we're seeing. So if there's maybe 400 species in Japan, there's at least 120 new species out there at least. And this is where they're all found, sort of scattered throughout with the archipelago. Tokyo, that's actually Tokyo Prefecture. That's the Ogasawara Islands that I'm hoping to visit with James in September. So again, there's a lot of travel, but it is a good excuse to see a lot of Japan as well. And yeah, look, and finally the acknowledgements. My main collaborators, Tom, Pete, Andrea Grottoli, sorry, and yes, and then a whole lot of people in Japan that have helped me get permits and organise travel, and yeah, and of course my host at Sissoko, Sakihari. Thank you.
Cool talk. Sorry I missed it at Uday. I'm happy I get to see it here. So super simple question, but maybe there's no answer. I'm not sure. So one of the other things in Veron's book that he talked a lot about was reticulate evolution and the long pelagic larval duration, the PLD of corals, and why this could be a mechanism for all the species evolving quickly. So what the UCE data is showing is a lot more endemicity than has previously been thought. So, and your background is reproduction originally, or one of your backgrounds is reproductive biology. So is it the PLDs not as long as people thought, or are there just a lot of reproductive barriers in there that is causing all of this endemism, or what's going on? That's the only thing that I really want to know.
Yeah, well, a couple of things. We're hoping that UCS will allow us to test these concepts of reticulate evolution in a way that hasn't been done before.
Yeah.
And secondly, where did this idea of corals being so widely spread come from? Yeah, it's interesting. PLD, my time as a larval biologist, is a rotten way of measuring how far organisms can spread. So, for example, in my PhD, I kept Acropora valida larvae alive for 103 days.
Yeah.
But only 5% of them could settle after that time.
Yeah.
So, they have a very long curve.
So,
the sort of median settlement for the median point of settlement for an Acropora species that can even, some of them can last for 100 days is probably 7 to 10 days. So most coral larvae don't disperse nearly as widely as we think. And yes, there must be, my guess is that there are also significant barriers to dispersal.
So those random things on the tail, or the random typhoon event that drags larvae all over the place, are really, evolutionarily speaking, usually inconsequential, is what you're sort of saying?
Yeah. I just think long distance dispersal is not nearly as frequent.
Well, basically zeals for sure, right?
Yeah. And yet the other aspect of reticulate evolution is obviously hybridization. Yeah. And again, I think it's not the major evolutionary force that people think it is. Again, it's one of my PhD students is using the UCE data to test this.
Yeah.
And again, in my experience in the lab, I've been trying to produce hybrids for 20 years, and I haven't been able to do it. Everyone else seems to be able to do it, but yeah. So I think so. Corals aren't, they don't disperse widely. I think they've got really much more specific habitat requirements that people have realised. The phenotypic plasticity was a good idea at the time, and obviously it's real, but not nearly as
much as
as people thought. So yeah.
Super cool. It's good to know there's tons of work left. Thanks.
No, well, that's it. There's no shortage of work.
No kidding. Right on. Thank you.
Michael?
I had a question about, have you looked up the symbiotes also? And
No.
okay, because
I think we can. I know Pete is exploring whether or not we're picking that up with the targeted bait set. So potentially down the track, but yeah, not yet.
because that'd be really cool because people have always said they're so diverse and maybe there's more patterns or.
Yeah, indeed. I mean, again, I suppose the issue with the real breakthrough here with the corals is a molecular method that allows species discrimination. I'm not sure if they're there with symbionts either, are they? So maybe they are, but again, I think that's what held the field back for a long time. All those sorts of questions about specificity and yeah, you can't ask them if you don't know what a species is. Yeah. David?
Andrew, nice talk. So if taxonomy moves one death at a time, perhaps you're trying to give Charlie a heart attack, or waiting for Charlie and Carden to move on, essentially.
Well, I think, I mean, realistically, I think they have both moved on, but
So my actual question is, the thing that worries me about this project is that, what are you calling a species in terms of molecular variation? I kind of, when we looked at the West Australian Acropora digitifera, you compare it, we did some population genomics on several populations. Those sequences at a whole genome level are remarkably similar to there's not much difference between those genome sequences and the Japanese Acropora digitifera, the OS digitifera sequence. And yet, talking to Tom and yourself, they're supposed to be radically different in terms of, so what are you calling, what's the variation that you take in UCE data to represent a species?
Well, I mean, again, this is my role is sort of to compare them to the type material and the ecology and yeah. So my understanding with the digitifera is that the species delimitation methods they use are what are pulling those different clades apart. So yeah, I mean, most of the firm decisions we're making in terms of what's species level difference and what's just geographic variation within a population, we're leaving to the algorithms, to at least do it quantitatively. And my understanding with digitifera is that the various different species delimitation approaches they're using, pulling them all apart, and that's certainly what's happening in tenuous, and certainly what's happening in the hyacinthus clade. And that's one line of evidence, and it's also looking at the morphology. But and again, there's still large areas of the tree where we can't make adequate decisions. That's why we're calling them primary species hypotheses. And again, you might need non-tree approaches to distinguishing the species, like Jean-Francois' haplowebs. And as for a definition of a species, I think we've deliberately tried to avoid it. It probably differs in different parts of the tree, where we've got solid molecular information, we run with that, where we don't, like, one of my PhD students was looking at Posloporeides and Cylindrica and Lutea, this big, massive coral bommies and this tiny little branching species. The RADSEC data can't distinguish between those two species that are obviously ecologically different. So in that situation, we tried some breeding trials. So I think there's so many different definitions of species that that we've deliberately avoided it today.
I have a question, Andrew, about you mentioned 30% endemism, but that's in general, or you're talking about specifically in Okinawa? Or just anyway?
Yeah, it seems to be, wherever we go,
Okay.
about 30% of the species are undescribed. And so, Lord Howe, yeah, obviously, depends how big your region is, but that's, Tahiti, Fiji, the Great Barrier Reef. It's going to vary enormously. Like somewhere like the Red Sea, it's probably 100% endemicity. Certainly in terms of the Acropora, there'll be a little bit of spillage out into the Western Indian Ocean. But areas like the Gulf, where the environment is so fundamentally different, they're going to be higher levels of endemism, and more isolated places, Hawaii, it's going to have higher levels of endemism as well.
James?
Sorry, one more.
Vincent?
Okay.
Sorry.
Okay, one super quick question. So of the scleractinians you've examined, you mentioned in your talk that you found nothing really in common between, say, the Red Sea and the Great Barrier Reef or the Indian Ocean. Are there any species out there that are all over the place generalists, or are you seeing this pretty consistent with just about any group of scleractinians you're looking at?
Well, we've only looked at the Acropora and the Montipora.
Okay.
And we haven't looked at the Montipora very closely.
Okay.
So within the Acroporidae, there's very few widespread species, right? And again, we need to be careful because we haven't been randomly sampling assemblages where we go. We target the topotypes and we target stuff that looks interesting. So there's by no means is our sample the sort of random sample that you would want to be able to put accurate estimates of overlap. But we really are seeing very few species. And interesting patterns too, like the fauna in Japan. The Acropora fauna in Japan seems to be more similar to the fauna in Western Australia than it does to the Barrier Reef. Now, that makes perfect sense when you think of patterns of connectivity.
And so these results in terms of endemism, do you think it's specific to anthozoans, or do you think you can apply this across to fish or other groups out there, other reef animals out there?
No, I mean, again, I think people know their fish a lot better than people have known their corals. Look, to be honest, I don't know whether it'll apply in other genera. I think the Acropora are quite unusual. They grow quick, they breed early. And so I think they're going to be very different to groups like the Porides, which grow slow and breed very late. So the generation times in the Porides are on the order of 20, 30 years compared to 2 or 3 years. So I think the patterns are going to be, there may well be some really widespread Porides. And certainly Tullia's data suggests that, within the limits of the genetic resolution she can get, she is seeing species in common between the GBR and the Red Sea. But I'm not convinced that that she's got the necessary resolution. But I think you would expect that our results might apply to other genera that are grow quickly and breed early. So Postloporids, Montipora, things like that. But the Merriolinids, Porides, don't know.
Cool. Looking forward to it. Thanks.
Sorry for a question that may be a bit seems to a bit provocative, but I am not a specialist at all of corals. I'm working on anemone fish, but I found some sort of a paradox in your talk where you told us, and I was quite convinced that the taxonomy is poorly known and there are many undescribed species, simply on the level of my small group of fish, 28 species described. There are some species with a wide distribution like Amphiprion clarki, where we effectively think that maybe it's more complicated. But in the same way, you told us also that you do not very well know how to define a species. So how could you discuss about species identity when in fact you don't know what is a species? So does it not mean that you should use another concept like, I don't know, some sort of molecular ecotype, or I don't know how to call it, but I found a paradox here.
Yeah, no, look, fair enough. That's why we do talk about the primary species hypotheses, and I have seen in the literature people talking about genetic species hypotheses. So and so that's essentially what I'm talking about. Amongst our 272 primary species hypotheses, not all of them are going to be good species. Not all of them are going to stand the test of the more quantitative approaches, like the species delimitation methods. No, absolutely, and I agree. I probably should be more specific about that. That we're still there's a lot of uncertainty in these data.
Well, I think we are in a situation which is very often the case in biology, where there is a very complex continuum, and we want to make limits where there is no, and so it makes our life very complicated. But so I must say I don't know how to handle it myself.
No, thanks.
Okay. Any other question? Jacopo, there are some questions from the Zoom, no?
No.
Sorry. Go ahead.
Very fundamental question. And then the project itself is very interesting. And then so what do you think the column which one is important to identify the species? Basically morphology or like a combination of the molecular? And I just wondering because if you identify the different and the polar species, it seems similar from the different locations, but the molecularly quite different. So which one you can trust?
It's again, people talk about multiple lines of evidence, but when those multiple lines of evidence are in conflict, how do you make decisions? Again, I suppose one thing we're trying to do as an ecologist is sort of reverse engineer from all these different lines of evidence some characters that we can use in the field. But it's going to differ for different groups of species. There's just no one character or one approach that's going to work in at every opportunity. In some groups, color will work really well. In other groups, there will be obvious morphological distinctions. In some groups, there'll be cryptic species that can only be distinguished with molecular techniques or other techniques. So there's sort of no one general rule or character. Whatever works.
Okay, so then thanks again, Andrew, for this. So Andrew will be still around today at Hoist. If you want to talk to him, we'll be here in Lab 4. Just come to see me, and he's happy to have a chat with you guys. Thank you.
Thank you.